Hello everyone, my name is Thalia, and today I'll go over the literature critique for the paper Programmable Editing of a Target DNA Base Without Double-Stranded DNA Cleavage, published in 2016 by the researchers of Dr. David Liu's lab. So every day, 30 trillion cells in your body would give rise to billions of mutations. And before it freaks you out, most of those point mutations are harmless. However, if they are early enough in your developmental stages, and if they are at important sequences, then they could cause human genetic disorders. In fact, most of the human genetic disorders are caused by point mutations, more than half of them. And most of the common ones that you might know include sickle cell anemia or cystic fibrosis, and many more like muscular dystrophy, progeria, and Tay-Sachs disease. So there's a need to address diseases caused by point mutation, and the most desired outcome is to convert an AT point mutation back into a GC. However, for this paper, we would focus on converting a CG base pair back into a TA base pair. And from what we have learned with the CRISPR-Cas9 discovery in 2012, it can cause a double-strand DNA break and then can be followed by two different repair mechanisms of the non-homologous end joining repair mechanism and the homology-directed repair mechanism. So for the non-homologous end joining repair mechanism, it would introduce random insertion or deletion of DNA at the cut region of interest. So as you can see from this chart here, it's very random, and most of the time it would deactivate or disrupt the function of the gene of interest. And that is not what we want with point mutation disorders, right? We would want to have a conversion of the unhealthy form or the disease-inducing form back into the healthy form, not to further disrupt whatever mutations that we have already had at this cut region. And with homology-directed repair, there's indeed a way to precisely introduce a gene of interest at the cut region. However, this process is not applicable for many cells because it's restricted to cells that have high replication rate. So the brain or the cardiomyocytes cells in your body would not be able to employ this method. So all of this is just to say that we need to develop a method that can replace point mutations precisely without causing DNA breaks. Okay, so the development of a specific guide RNA programmed cytosine converted to uridine base editor generation 1 without causing double-stranded DNA breaks in vitro was a solution to this challenge. So what the researchers of this lab cleverly did was that they used a deactivated Cas9 protein so that they turned off the nuclease activity with the point mutation of aspartic acid and alanine to prevent it from causing a DNA cleavage, but it can still bind to a targeted DNA sequence. And then they also use a cytidine deaminase that can convert deaminase, a cytosine, into a uridine that have a thymine base pairing properties. They actually screen out different cytidine deaminase in rat, in humans, and in fish, and then they chose to proceed with the rat version because it's had the highest deaminase activity. They also tested out different linkers to connect the cytidine deaminase and the Cas9 protein with different lengths and composition. And most notably is the 16 amino acid X10 peptide linker. And the green one is the guide RNA and DNA complex that I'll go over in a little bit. So the mechanism of action is that the Cas9 protein can bind to the targeted double-strand, the DNA of interest. And then the non-complementary strand to the guide RNA would be where the cytidine deaminase binds to, and the enzyme would then induce a change to deaminase the cytosine into a uridine. And because uridine has the thymine base pairing property, after a few replication cycle, you would permanently change a CG base pair into an AT. And remember that from now on, I would refer to the base distal to the BAM sequence. So this is a BAM sequence, and this is a base distal to the BAM sequence as position 1. Okay, so the authors of this paper actually described CRISPR 2.0 version of the base editing method as a pencil eraser analogy compared to a scissor and glue analogy of CRISPR 1.0 version, because you can literally go in and rewrite whatever base that you want at the targeted sequence. So in this case, it would be precisely changing a cytosine into a uridine or thymine. But then you may ask, how precise is that eraser? So there are three major things that we're going to go over today because the paper addressed all these issues very eloquently. One is the specificity and off-target effects of this base editing method. And two is how it bypassed cellular repair mechanisms. And three is its clinical applications in vitro and in vivo. All right, so first of all, regarding the specificity and off-target effects, as you may know, with CRISPR-Cas9, it does not bind to just one nucleotide. It binds to a fragment of DNA. So then how precise is this complex, this base editing complex, able to just go in and edit out the cytosine that we want instead of any other cytosines of the fragment DNA that we're targeting? So what they did was that they carried out a deamination assay and added in BE1, a base editing generation 1. And each lane over here is labeled according to the position of the target cytosine within the protospace. And remember that the base distal from the BAM is position 1. And the result was that there was maximum editing efficiency achieved when the target C is at or near position 7. And there's a window of activity of 5 nucleotides from around 4 to 8 from the distal end of BAM or position 1. So now that we know that the maximum efficiency is at position 7 away from the distal end of the BAM, they carried on another experiment to study, okay, what bases around, like neighboring to the target position 7, do we need to improve the specificity and efficacy of the base editing generation 1. So they assay BE1 ability to edit one single C at position 7, and then they swap the neighboring bases with A, G, and T, respectively. So as you can see here, editing efficiency was independent of the sequence context unless the base immediately 5' to position 7 was a G, which would substantially lower the efficacy. And the base editor 1 best activity followed the order of TC and then CC and then AC and then GC. They also concluded that off-target base editing is only from off-target Cas9 binding, and base editing is retained to a 5 nucleotide window position. So then the second question you may ask is, what about cellular repair mechanisms, as this will be important in vivo studies? So in the cells, there are two major mechanisms to repair the U that we just worked so hard to edit from the C. The first one is that it just employ uracil DNA glycosylase that just converts the uridine back into a C. And the second repair mechanism is that it can induce a nick on the edited strand. And because of that nick, the cell would know that, oh, there's some error with that strand, and it's gonna fix the U and convert it back into a C. So they cleverly developed two generations, BE2 with a uracil DNA glycosylase inhibitor that basically blocks the human UDG activity, and fuse that with the first generation BE1 to create the second generation base editor BE2 with an improvement in efficiency threefold that of BE1. And both of them had very low indel formation or like random deletion of DNA rates of less than 0.1%. And then they also developed the third generation with the mechanism to bypass the mismatch repair nicks, and they indeed induce a nick on the unedited strand. So they would replace a G to an A, and that would eventually favor having a TA base pair instead of a CG base pair originally. And this would overall improve a total efficacy to potentially higher than 50% instead of just the maximum 50% of only one strand. So then they also compared the base editing efficacy between different base editing generations on human cells. So as you can see, that base editing generation 1 has very low conversion rate from C to T because it doesn't have way to bypass the cellular repair mechanisms. 2 was better with a UDI, and then the real deal was base editor generation 3 with both UDI and way to bypass the mismatch repair by inducing a nick on a non-edited strand. And they all have very low indel formation rate. So they also carried out clinical test experiments to see how it can be clinically applied. So they tested the base editing method to correct 2 disease-relevant mutations in mammalian cells, and they very cleverly inserted human mutations into animal models, namely mouse models. So I think this is why it deserves to be published in Nature. It just one step further from doing all these experiments in human cells and taking it into animal models infected with human genes. So they basically targeted the arginine position 112 and 158 and tried to replace it with cysteine to produce ABOE3, and this would significantly lower the risk factor of Alzheimer's disease. And when they added in BE3, they were able to show a efficacy of converting a C to a T of 58 to 75% of total DNA sequencing reads. And they also did it with a similar mutation that caused cancer. And they were able to observe a correction of tyrosine to cysteine at position 163 with the efficacy of 3.3 to 7.6% compared to only less than 0.1 or not at all efficient when we use the Cas9 and donor DNA method. So some of the positive and negatives of the paper is that one, it offers a very powerful tool to precisely modify a point mutation, changing a C into a U without causing DNA breaks. It also had very well-designed experiments to study the specificity and effects of narrowing bases around a targeted cytosine. And they also set out to optimize the editing efficiency of the first generation base editing method in human cells with the urine glycosylase inhibitor with the second generation and way to bypass the mismatch repair with the third generation. And there was a clear transition from in vitro analysis with just human cells to using animal models like mice with infected human genes and mutations. And the clinical applications were done with 2-point mutation diseases, and both showed superior base editing efficacy compared to the traditional method of using CRISPR and homologous directed repair. In the supplementary files, they also included a long list of potential candidate point mutation diseases that can potentially employ this technique as therapeutics. A negative point that I'd like to point out is that the paper focused on providing a solution to fix CG to AT point mutation. But from what I've shown on the first slide, most point mutations occurring in human require modification from an AT to a CG. So more discussion on if a similar mechanism to carry out a ladder-based edit should be made to address some just interesting questions in the future. And they also mentioned a slightly broad window activity of 5 nucleotides. So how would it transfer to clinical meanings, clinical applications? Because it is kind of precise, but it's also there's also like some kind of leeway of 5 nucleotide window activity. So that's something to consider. And the paper also mentioned inducing a nick on the non-edited strand to convert a G to an A to favor the original edit. How easily is the procedure carried out? Is it also mediated by Cas9 so that mismatch repair enzyme knows specifically where to nick? And my suggestion to the authors is that when they talked about how the off-target binding affinity was only due to Cas9 binding off targets, however, the window activity, again, of 5 nucleotides can be quite broad. So more discussion on the specificity and clinical applications of this broad window base editing activity should be emphasized for further studies. Furthermore, they only carried out an experiment to study the most optimal position to edit the cytosine at position 7 with only the first generation. So similar experiments should be carried out for generation 2 and generation 3 to see if position 7 is still the best target position. The paper also mentioned inducing a nick on a non-edited strand, but they should discuss a little bit more on how easy it is to perform and if they also employ Cas9 to target desired sequences. And then they also should discuss the likeliness of developing similar approach to edit AT back to CG, which is the most common disease-inducing point mutation in human. And if there are major challenges to why this is not possible, then it should also be included on there just so that other authors who are interested in further developing this method know where is the starting point. And these are some of my references. Thank you very much.